Hey students, this is your Nandini ma'am. Today, in our audio chapter, we are going to start cell function. So let's get started. The development of technologies to view the internal structure of cells enabled scientists to determine the different types of cells and the structures they possess. With this knowledge, our curiosity about how cells function grew. Further development of technologies led to the study of the biochemical processes of photosynthesis and cellular respiration, which are carried out by cells in their internal environment to ensure the efficient and successful functioning of the organism. In order for these biochemical processes to occur, cells must obtain the materials necessary from their external environment and must be able to remove any wastes produced from their internal environment and coordinate these activities. Coordination of the biochemical activities of the cell to ensure efficient cellular metabolism is the responsibility of the nucleus. The cell membrane controls the movement of cellular requirements from the external environment of the cell into the cell and the removal of cellular products and wastes from the internal environment of the cell into the external environment. Movement of materials in and out of cells. For any cell to function effectively, it must interact with its surrounding environment and with the cells that surround it. Substances required by cells for their functioning need to move into the internal environment of cells, and waste substances and cellular products need to pass out of cells into the external environment. These substances move from the internal environment to the external environment by passing through the cell membrane. Substances needed by cells are gases, nutrients, and water, the main solvent in cells. Mineral salts dissolved in the water are also required. Substances that must leave cells are wastes such as urea, uric acid, and excess carbon dioxide. They could also be products secreted by cells that may be needed to coat the outside of cells and may pass to the other cells. In both plant and animal cells, the cell membrane is in direct contact with the cytoplasm inside the cell. The cell membrane is selectively permeable or sometimes referred to as differentially permeable, meaning it controls what passes across it. The membrane controls the passage of water and other molecules into and out of living cells. In contrast to the selectively permeable nature of a cell membrane, the cellulose cell wall in plant cells is permeable. It is a non-selective boundary that allows water and most molecules to pass freely inwards or outwards. The movement of these molecules is restricted only when they come into contact with the cell membrane. To understand how the cell membrane controls and regulates the movement of substances, biologists examine the structure of the cell membrane and relate this to its functioning. Movement of molecules across cell membranes. The permeability of a cell membrane to a molecule depends on the molecule's size, electrical charge, and lipid solubility. Some molecules can move across membranes quickly. Larger molecules have more difficulty moving across membranes. Water-soluble molecules have difficulty penetrating a membrane, whereas lipid-soluble molecules do not. This is due to the phospholipid bilayer structure of the cell membrane. The lipid tails impede the progress of hydrophilic molecules and hence the movement of lipid-soluble molecules such as urea and ethanol. Electrically charged molecules such as sodium and potassium ions are not very soluble in lipids and therefore have low membrane permeability. Neutral molecules such as carbon dioxide and oxygen gas are soluble in lipids and have a high permeability. Although water is a polar molecule that is not lipid soluble, membranes have pores that make them highly permeable to it. Water moves through special tiny hydrophilic pores in the membrane called aquaporins by a process called osmosis. Molecules that have low permeability rely on carrier proteins to transport them across membranes in cells. This will be dealt with in more detail later in this chapter. Key concepts. Substances move from the internal environment of the cell to the external environment by passing across the cell membrane. The cell membrane is selectively permeable and controls the movement of substances into and out of the cell. Gases, nutrients, water, and ions are required by the cell, while wastes and cellular products need to be removed from the cell. The permeability of a membrane to a particular molecule depends on its size, electrical charge, and its lipid solubility. Plant cell walls are permeable to most substances. Diffusion. The movement of materials into and out of cells takes place either passively or actively. Passive movement requires no energy input and includes the processes of diffusion and osmosis. Diffusion is the net movement of any molecules from a region of high concentration to a region of low concentration of that substance until equilibrium is reached. Equilibrium is reached when that there is no net movement of molecules in either direction. The molecules move equally in each direction. This process does not require an energy input. For example, if someone sprays a burst of perfume in the back corner of a laboratory, the students closest to the corner will smell it quickly. Students at the front of the room will not smell it until some time later. The perfume molecules will gradually move from the back of the room where it has the highest concentration to the front of the room where it has the lowest concentration. This will continue until the concentration is the same throughout the whole room, that is, it is an equilibrium. The perfume is said to have diffused throughout the laboratory. No energy input has been required for this process to occur. So movement from a high concentration to a low concentration is described as movement along a concentration gradient. Molecules moving down a concentration gradient can be linked to the movement of rocks rolling down a hill needing no energy input. The rate of diffusion changes depending on the concentration gradient. If there is a greater difference in the concentration of substances, the concentration gradient will be steeper and diffusion will occur faster. Diffusion can also speed up or slow down depending on the temperature. Heat increases the rate of diffusion because the kinetic energy of the particles increases. Investigation 3.1. An investigation to model the process of diffusion through the cell membrane. In this investigation, you will model diffusion through the cell membrane using semi-permeable dialysis tubing to represent the selectively permeable nature of the cell membrane. The starch solution is milky white in color and will be used to represent the contents of the cell. The iodide ions in the yellow iodine potassium solution represent a cell requirement that will readily diffuse through the cell membrane. Aim: to model the process of diffusion through the cell membrane. Materials: 2 into 800 ml beakers, starch solution, iodine potassium iodide solution, 2 into 20 centimeter section of dialysis tubing, filter funnel, fine string or cotton thread, 500 ml measuring cylinder, 2 into glass rod, scissors. Risk assessment. What are the hazards? What risk does this hazard pose? How can you safely manage this risk? Iodine potassium iodide solution. Irritant to skin and eyes. Poisoning if ingested. Irritant if inhaled. Do not ingest the chemical. Do not use in confined spaces. Wear safety glasses. Broken glass. Use cuts to the skin. Take care when using glassware. Method. Use one of the beakers to soak the dialysis tubing in water for 5 minutes. Fill the other beaker 3/4 full with tap water. Rub the end of the tubing between, add 10 drops of iodine potassium iodide solution and mix. Remove the tubing from the water and underwater. Rub the end of the tubing between your fingers to separate the sides. Tie one end very securely with string. Measure 30 to 40 ml of starch solution into the measuring cylinder and pour into the tubing using a filter funnel. Tie off the top end of the tubing very securely with string, leaving a length of string to secure the tubing to the glass rod. Wash the outside of the tubing. Tie the end of the string to the middle of the glass rod. Place the rod across the top of the beaker to suspend the tube containing the starch solution in the water plus iodine potassium iodide solution. Ensure that the tube is fully submerged. Leave undisturbed for 15 to 20 minutes. Observe and record any color changes that occur. Use the other beaker to set up a control, leaving out the iodine potassium iodide solution. Results. Record your observations in a table like 3.1 for the experiment and control discussion. Copy and complete table 3.2, which identifies the correlation between your model of diffusion and diffusion across the cell membrane. Using the results obtained, copy and complete table 3.3, which indicates the initial and final concentrations of starch molecules and iodide ions both inside and outside the dialysis tube. Use terms such as highly concentrated, none present, decreased concentration, increased concentration, and no change. Describe evidence from your investigation to support your conclusions in Table 3.3 about the change or no change in the concentration of iodide ions, starch molecules. Explain how this investigation has modeled the process of diffusion through the cell membrane. Use the term concentration gradient and control and refer to the selectively permeable nature of the cell membrane and to the size of the molecules. Discuss the benefits and limitations of the model. Write a conclusion to link your results to the aim of the investigation. Diffusion across a cell membrane. Small uncharged particles such as carbon dioxide and oxygen move easily through the cell membrane by simple diffusion. These particles pass between the phospholipid molecules from a region of high concentration to a region of low concentration. The concentration gradient is usually maintained for the diffusion of oxygen molecules because oxygen is removed for use in the cell. Oxygen is continually used in the process of cellular respiration, keeping its concentration low inside the cell. This promotes the diffusion of oxygen from outside the cell where it is in high concentration. Facilitated diffusion. Relatively large molecules such as glucose and amino acids and charged particles such as sodium and chloride ions do not readily pass through the phospholipid bilayer. They require certain proteins called carrier proteins or channel proteins in the cell membrane to assist them in diffusing into the cell. This process is called a facilitated diffusion. As shown in figure 3.1, the carrier proteins bind to molecules on one side of the membrane. They then change shape and release the substance on the other side. The direction of movement, whether into or out of the cell, depends on the direction of the concentration gradient. Small ions such as sodium ions diffuse rapidly through the cell membrane from a high ion concentration to a low ion concentration via narrow passages called channel proteins. These channel proteins are specific for particular ions. Key concepts. Diffusion is the movement of particles from a region of high concentration to a region of low concentration until equilibrium is reached. At equilibrium, there is no net movement of particles in either direction. Diffusion does not require the input of energy. Diffusion occurs faster with a higher temperature or a steeper concentration gradient. Small, uncharged molecules such as oxygen and carbon dioxide will diffuse easily across the cell membrane. Facilitated diffusion allows larger molecules and small electrically charged ions to diffuse across the cell membrane aided by carrier or channel proteins. Osmosis. Osmosis is a special type of diffusion. It is the net movement of solvent molecules from a region of high solvent concentration to a region of low solvent concentration through a semi-permeable membrane. When water is the solvent, just like diffusion, the movement of water occurs along the concentration gradient and does not require an energy input. Water is very important to living things. It is the medium in which many of the biochemical reactions in cells occur. Water helps keep cells in shape, so it forms the fluid that bathes tissues and it also transports materials in solution. A solution is formed when a solute dissolves in a solvent. The amount of solute dissolved in a given quantity of solvent determines the concentration of the solution. Water is the most common solvent in a solution. A concentrated solution contains a large amount of solute in relation to the amount of water, so the water is said to be in low concentration. A dilute solution contains a small amount of solute in relation to the amount of water, and the water is said to be in high concentration. Osmosis is the process by which water moves through the cell membrane. Because water is not lipid soluble, the movement is not directly through the lipid bilayer. Water moves through special tiny protein channels in cell membranes called aquaporins or water pores. When water is more highly concentrated outside the cell than it is inside the cell, water will move by osmosis through the selectively permeable cell membrane into the cell and the cell may swell up. Alternatively, if the concentration of water is lower outside the cell than inside, water will move out of the cell by osmosis and the cell may shrink. The pressure created by water moving across a semi-permeable membrane due to osmosis is called the osmotic pressure. The more water that moves across the membrane, the higher the osmotic pressure created. If the fluids inside and outside a cell are of equal solute concentration, the external solution is said to be isotonic to the cell contents. Water molecules jostle on both sides of the membrane, moving in both directions equally. When cells are surrounded by a solution that contains a lower solute concentration than their cytoplasm, the external solution is said to be hypotonic to the cell contents. Net movement of water molecules will be through the membrane into the cells. The reverse applies if the cells are surrounded by a solution of higher solute concentration. The external solution is hypertonic to the cells and net movement of water molecules will be out of the cells. Investigation 3.2. An investigation to model the process of osmosis. In this model of osmosis, the semi-permeable dialysis tubing represents the selectively permeable cell membrane. The sucrose solution represents the cytoplasm of the cell, and the distilled water represents the extracellular fluid. Aim to model the process of osmosis. Materials: 3 into 600 ml beakers, distilled water, sucrose solution, fine string, 2 into glass rods, 2 into 20 centimeter strips, dialysis tubing, 25 ml measuring cylinder, 100 ml measuring cylinder, filter funnel, marking pen, plastic cling wrap, digital camera, optional. Risk assessment: broken glass, injury to body. Take care at all times when using glassware. Method: cut 2 pieces of dialysis tubing to 20 centimeter lengths and soak in a beaker of water for about 5 minutes. Tie one end of each piece of tubing securely with a piece of string, leaving the other end untied. Run the untied end of the tubing under water and rub it between your fingers to open it. Using a filter funnel and a measuring cylinder, pour a measured volume of sugar solution into the tubing so that the tubing is 2/3 full. Record the exact volume of sugar solution used. Tie the top of the tubing with string, attach it to a glass rod as shown in figure 3.12. This is your experimental apparatus. Repeat the previous 2 steps, this time filling the tubing with distilled water instead of sugar solution. This is your control apparatus. Using a measuring cylinder, fill each beaker about half full and record an accurate measurement of the volume of water in each. Suspend the experimental and the control apparatus each in a beaker of distilled water as shown in figure 3.12. Ensure that the tied ends of the dialysis tubing are just above the distilled water in the beaker to prevent leakage. Do not have the tubing too far out of the water because evaporation may occur and this will interfere with the accuracy of your results. Mark the level of water in the beaker with the marking pen. Cover the top of the experiment with plastic wrap to prevent the water in the beaker from evaporating. Leave the apparatus to stand overnight. Measure the volume of the solutions in each bag after they have been left overnight. Record the volumes of the solutions in each dialysis bag and each beaker at the end of the experiment. Compare this with the volume recorded at the start of the experiment. Also record the number of hours the experiment was left standing. Observations such as the change in the fullness of the dialysis tubing and the changing water level should also be recorded. Results. Your results will be both quantitative and qualitative. Draw up a suitable table to record your measurements. Describe any change in water levels in both the beakers and the dialysis tubing. Diagrams are often useful tools to aid descriptions. Discussion. Identify each of the following in your investigation: the independent variable, the dependent variable, the controlled variables. Why was the dialysis tubing with distilled water used with the beaker with distilled water? Complete table 3.4, which identifies the correlation between your model of osmosis and osmosis across the cell membrane. Describe how your investigation has modeled the process of osmosis. Outline any limitations of your model. Discuss improvements that could be made to your model of osmosis. Conclusion. Write a conclusion to link your results to the aim of the investigations. Key concepts. A solution is formed when a solute dissolves in solvent. A concentrated solution has a high concentration of solute and a low concentration of water. A dilute solution has a low concentration of solute and a high concentration of water. Osmosis is the process by which water moves from a region of high concentration of water to a region of low concentration of water. Osmosis requires no energy input. The more water that moves across the membrane, the higher the osmotic pressure created. Isotonic fluids inside and outside a cell are of equal solute concentration, no net water movement. Hypertonic, a solution of higher solute concentration that surrounds a cell, so net movement of water molecules will be out of the cell. Hypotonic, a solution of lower solute concentration that surrounds a cell, net movement of water molecules will be into the cell. Osmosis in animal cells. The cells of unicellular eukaryotes are surrounded only by a cell membrane. Hypotonic solutions such as fresh water pose a problem because water moving into animal cells by osmosis can cause the cell to swell and eventually burst the cell membrane, killing the organism. Cells in most animals are not directly exposed to the external environment and are bathed in isotonic extracellular fluid. This means that cells can function efficiently because water diffuses equally in both directions, resulting in no net movement of water into and out of cells. The water concentration in animal cells needs to be kept constant to coordinate biochemical reactions. Osmosis in plant cells. Unlike animal cells, plant cells will not burst when soaked in fresh water, even though water moves into the cell by osmosis. Plant cells usually have large fluid-filled vacuoles and firm semi-permeable the cell walls that surround the cell membrane. Plant cell vacuoles contain cell sap that has high concentration of solutes and therefore a low concentration of water. When a hypotonic solution surrounds a plant cell, the water molecules move by osmosis into the vacuole. This causes the vacuole to swell and pushes the cell membrane outwards against the cell wall. The tough cell wall prevents the cell from bursting. When the cell wall stretches as much as possible, no more water can enter the cell and the cell is said to be turgid. In this state, the osmotic pressure inside the cell is equal to the opposing pressure exerted by the cell wall. If the plant cells were to be placed into a hypertonic solution, the water in the cell would leave the cell by osmosis. The vacuole would shrink and cause the cell membrane to move away from the cell wall in a process called plasmolysis. Active transport. Just a second. Yeah. Active transport is the movement of molecules from a region of low concentration to a region of high concentration and requires the input of energy. This movement goes against the concentration gradient and involves movement across a cell membrane that has receptors for the molecules. Diffusion and osmosis both rely on a concentration gradient to direct the passive flow of substances from regions of high concentration to regions of low concentration. Sometimes in living things, a chemical may need to be moved against the concentration gradient, such as when kidney cells reabsorb glucose and amino acids so they are not lost in urine. Active transport requires a carrier protein that spans the membrane to actively move chemicals from a low to a high concentration utilizing cellular energy. Key concepts. Active transport is the movement of molecules from a region of low concentration to a region of high concentration. Active transport moves against the concentration gradient and requires the input of energy. Endocytosis and exocytosis, transport of large molecules. Sometimes particles that are too large to move through the cell membrane by either diffusion or active transport need to enter or leave a cell. Endocytosis. When a large molecule has to be moved into a cell, the cell membrane can change its shape to surround the molecule and engulf it by the process of endocytosis. If a solid particle is engulfed, the process is termed phagocytosis. Sometimes fluid is engulfed and the process is then called pinocytosis. One example of phagocytosis occurs when a unicellular amoeba feeds on a smaller organism. The amoeba changes shape by sending out membrane projections filled with cytoplasm that surround the prey. When the cell membrane of the projections meet, the membrane fusion occurs. This results in the formation of a vesicle, which then stores or transports the material within the cytoplasm. Pinocytosis occurs when the cell membrane engulfs a drop of extracellular fluid in much the same way as phagocytosis. Fat droplets found in the small intestine after a meal move into the cells by the means of pinocytosis. Exocytosis. Specialized animal and plant cells produce a variety of substances such as antibodies, neurotransmitters, and enzymes that have important functions elsewhere in the organism. These substances are contained within vesicles inside the cell. Cells also produce waste products that need to be moved out of the cell. Exocytosis is the process by which these substances are transported to the external environment of the cell. During exocytosis, a membrane-bound vesicle moves to the cell membrane, fuses with it, and then releases its contents to the exterior of the cell. The vesicle membrane becomes part of the cell membrane. Key concepts. Endocytosis moves large molecules that cannot cross the cell membrane into a cell. It requires the expenditure of energy. In endocytosis, the cell membrane changes shape and surrounds and engulfs the particles so that it enters the cell. Phagocytosis is the process whereby solid particles are engulfed by the cell membrane. Pinocytosis is the engulfing of fluid substances by the cell membrane. Exocytosis involves a membrane-bound vesicle moving to the cell membrane, fusing with it, and then releasing its contents to the exterior of the cell. Factors affecting the exchange of materials across membranes. The characteristics of the cell membrane and the physical and chemical properties of the material will determine whether it can move through the cell membrane. Chemical factors. The chemical properties of a substance affect its transport across cell membranes. Many uncharged molecules such as ethanol can easily penetrate the cell membrane because they can dissolve in the phospholipid bilayer. Hydrophilic charged ions such as sodium and potassium cannot cross the hydrophobic center of the membrane. Channel proteins specific for each ion allow their movement through the cell membrane. Water is not lipid soluble and therefore cannot move through the hydrophobic tails in the cell membrane. Water moves through the membrane through the special aqua porins. Physical factors. The physical properties such as size and shape of the molecules affect the movement of substances across the cell membrane. Small molecules are able to diffuse easily between the phospholipids. Glucose and amino acids are examples of large molecules that use carrier proteins to move through the cell membrane. Very large molecules that need to be transported into and out of the cell are moved by the process of endocytosis or exocytosis. Concentration gradient. The relative concentration of the substance on either side of the membrane affects the rate of diffusion of that substance. If the concentration gradient is high, then the substance will diffuse rapidly. As the concentration gradient decreases, the rate of diffusion will be slower. In order to maintain a rapid rate of diffusion, cells need to maintain a higher concentration gradient. When the concentration reaches equilibrium, there will be no net movement across the cell membrane. Plant cells carry out a process called cytoplasmic streaming, which involves organelles and cytosol flowing around the cell in a circular movement. This enables the cell to maintain a steeper concentration gradient as materials that diffuse into the cell are rapidly moved to another area of the cell. Surface area to volume ratio. The surface area divided by the volume is called the surface area to volume ratio. This also affects the movement of substances into and out of the cell through the cell membrane. The surface area of a cell is the total area of the cell membrane that is around the cell. The volume of the cell is the space taken up by the internal contents of the cell. A cell needs to have enough surface area to supply its volume with requirements and remove waste. This ratio of a cell has a large impact on the movement of substances into and out of cells. A smaller cell has more surface area in relation to its volume, a higher surface area volume ratio. Think of the distance from the surface of a small cube to its center. This distance is much less than the distance from the surface to the center in a large cube. Therefore, a smaller size allows a faster movement of substances between the center and the surface of the cell. Anything that the cell needs can get to all parts of a small cell quickly, and wastes can be removed easily. This then allows the cell to perform at an optimum level of functioning. Figure 3.21 illustrates surface area volume ratio for simplified cubic cells, but surface area volume ratio is important for the movement of substances into and out of cell of any shape. A larger cell has a smaller amount of surface area in relation to its volume, a lower surface area volume ratio. This means the efficiency with which a cell obtains its nutrients and removes its waste is reduced as its size increases. A cell increasing in size reaches a point where the inward movement of essential substances and outward movement of wastes across the surface area by diffusion are not fast enough to service the increasing volume of the cell. When a cell reaches this size, if it is capable of dividing, it will often do so. For this reason, the individual cells tend to be very small. The shape of the cell also makes a difference to the surface area volume ratio. Spherical cells have a relatively small surface area volume ratio compared with cells of other shapes. Long, flat cells have a higher surface area volume ratio than a spherical cell with the same volume. Table 3.5 shows the surface area volume ratio for three hypothetical spherical cells. It demonstrates that the smaller cell A with a diameter of 1 mm has a higher surface area volume ratio of 6:1 than the larger diameter cells B and C. Cell B has a surface area volume ratio of 3:1 and cell C has a surface area volume ratio of 2:1. This means that for each unit of volume, cell A has 6 units of surface area, B has 3 units of surface area, and cell C has 2 units of surface area. It then allows or follows that the inward movement of essential substances and the outward movement of wastes across the surface area by diffusion would occur much more rapidly in cell A than in cell B and be slowest in the cell C. Cells often have specific features that ensure they have the largest surface area volume ratio. An example is seen in the root hairs that cover the root tips of most plants. The long, thin extensions of the single cells that form root hairs increase the surface area through which water and mineral salts can be absorbed. Key concepts. Surface area is calculated by finding the total area of the surface of the shape. Volume is the total space that a shape takes up. Surface area volume ratio is calculated by dividing the surface area of an object by its volume. The surface area to volume ratio of a cell will determine how efficiently substances move into and out of a cell. A high surface area volume ratio allows the most efficient movement of substances into and out of the cell. Large cells have a low surface area volume ratio. Small cells have a large surface area volume ratio. Long, flat cells have a higher surface area volume ratio than a spherical cell with the same volume. Cell requirements. Whatever or wherever life exists, it depends on a source of energy and a supply of matter. Cells need to obtain nutrients in the form of organic substances such as glucose, amino acids, fatty acids, and glycerol, nucleotides, and vitamins. Organic substances are synthesized by living things and contain carbon and hydrogen atoms. Cells also need inorganic nutrients such as gases, minerals, and water. These inorganic nutrients are part of the non-living world and do not contain carbon and hydrogen in long chains. The substances needed by living cells for their functioning are used in two main ways. as essential building blocks from which cells and living tissues are made, as a source of stored energy for the cell. Organic nutrients are the main supply of stored energy in the living things, but they are also used in the structure of cells. Inorganic nutrients are essential as structural parts of the cells and tissues. Inorganic nutrients. Some inorganic nutrients that occur in cells are listed in table 3.7. Water, oxygen, hydrogen, 90% of the protoplasm, cytoplasm, and nucleus. The transport medium in cells and in organisms, an important solvent for many molecules inside cells, the medium in which all chemical reactions in cells take place, water may be used in the reactions. Mineral salts, chlorides, nitrates, phosphates, and carbonates of sodium, magnesium, calcium, potassium, and ammonium, dissolved as ions in the cytoplasm and in vacuoles in plant cells. Assist all chemical reactions, are used in the synthesis of many macromolecules and body tissues. Sodium ions and chloride ions assist in water balance in cells and are essential for cell membrane functioning and the function of nerve and muscle cells. Gases, carbon dioxide, oxygen, dissolved in the protoplasm, used and or produced in chloroplast and mitochondria. Carbon dioxide, used during the process of photosynthesis, released as a product of aerobic cellular respiration. Oxygen, used by all living organisms during aerobic cellular respiration to release energy for cells to function, released as a byproduct of photosynthesis. Organic compounds. Every living cell requires large organic molecules as part of their structure and to maintain the biochemical processes involved in effective functioning. These large organic molecules are called biomacromolecules. There are four main types based on their chemical composition and structure: complex carbohydrates, lipids, proteins, and nucleic acids. Plants and other organisms carry out photosynthesis, absorb inorganic nutrients from the soil and the air, and use these to make their own organic nutrients. In contrast, organisms that do not carry out photosynthesis need to ingest food to obtain organic nutrients for their cells. Carbohydrates. Carbohydrates are a group of organic molecules made up of carbon, hydrogen, and oxygen atoms in the ratio of 1:2:1, giving the journal formula for carbohydrates as CH2O whole bracket X. Carbohydrates are classified as monosaccharides, disaccharides, and polysaccharides depending on how many monomers are linked. The product of photosynthesis, glucose, is a monosaccharide. Monosaccharides, simple sugars consisting of a single monomer, source of quick energy in both plant and animal cells. Disaccharides, complex sugars consisting of two monomers, like maltose, lactose, sucrose. Polysaccharides, complex molecules consisting of more than 5 and up to hundreds of monomers joined together. Starch, stored energy in the plant cell. Cellulose, structural part of cell walls, strength and support. Glycogen, stored energy in animal cells. Lipids. Lipids contain many carbon and hydrogen atoms with a few oxygen atoms. The fats and oils in the cells of organisms are typically composed of triglyceride molecules. These atoms are arranged as a glycerol molecule to which 3 chains of fatty acids attach. Lipids are relatively insoluble in water and have an oily, greasy, or waxy consistency. In cells, lipids have 3 important functions: energy storage. They store approximately twice the amount of energy as carbohydrates. Structural component of membranes, essential structural parts of hormones, which are chemical messengers produced by cells. Proteins. Proteins are made up of elements carbon, hydrogen, oxygen, nitrogen, and sometimes sulfur. These elements combine to form amino acids, which are the building blocks of proteins. There are about 20 different amino acids that can be put together in chains of up to 300 amino acids to form a peptide or polypeptide chain. Proteins are made up of one or more of these polypeptide chains twisted together into a particular shape. The DNA in the nucleus of the cell controls the sequence and arrangement of the amino acids, and this determines the type of proteins. Proteins have many roles in cells. They form structural components in cells and tissues. They are an important structural component of cell membranes. Some proteins have a functional role, such as enzymes, which control all the metabolic reactions in the cell, and hormones, which control the functioning of other cells. Proteins occur suspended in the protoplasm of cells or combined with other macromolecules to form an important structural part of all membranes within the cell. Plant cells can synthesize their own amino acids. Animal cells produce most of the amino acids required for protein production. 9 of the amino acids are called essential because they cannot be synthesized by cells in the body and must instead be sourced as in nutrients from the external environment, such as in food. Nucleic acids. Nucleic acids are very large biomacromolecules that contain the elements carbon, hydrogen, oxygen, nitrogen, and phosphorus. There are two types of nucleic acids: DNA, deoxyribonucleic acid, and RNA, ribonucleic acid. DNA is a double-stranded molecule that stores the information that controls the cell. It is the main chemical making up the nucleus. Small amounts are also found in mitochondria and chloroplasts. RNA is found in small amounts in the nucleus and in larger amounts in the cytoplasm. The building blocks of DNA and RNA are called nucleotides. Each nucleotide consists of a sugar, a subunit called a base, and phosphate. DNA nucleotides contain the four bases adenine, guanine, and cytosine and thymine. They also contain the sugar deoxyribose. DNA nucleotides are required by cells to make DNA during cell replication. RNA nucleotides contain the four bases adenine, guanine, cytosine, and uracil, and they also contain the sugar ribose. RNA nucleotides are required for the cells to make ribosomes and to make RNA so that cells can make proteins. Key concepts. The four main groups of biomacromolecules are carbohydrates, proteins, lipids, and nucleic acids. These four groups of organic compounds are made up of carbon, hydrogen, and oxygen atoms, but they are in different proportions. Proteins also contain nitrogen and sometimes sulfur. Nucleic acids are also containing nitrogen and phosphorus. Autotrophic organisms can build their own organic compounds, whereas consumer organisms must make their organic compounds from their food. Carbohydrates can be used as a source of energy for storage of energy and sometimes as a structural component of cell. Lipids are used for energy storage, structural parts of the cell membrane, and components of hormones. Proteins have a structural role in cells and tissues. Proteins such as enzymes also have a functional role. The two types of nucleic acids are DNA and RNA. DNA contains chemical information that controls the cell activities and the production of proteins. RNA assists in the manufacture of the proteins. This was part 1 of Cell, the chapter.